we have the capability to design proteins that do not exist in nature. With protein design, what we can do is shorten up the evolution.
The worst thing that can happen is that the resulting organism created in a laboratory will escape and create an epidemic of some sort.
Welcome to Science Forward, the podcast of the UN Secretary-General's Scientific Advisory Board. I'm Julia.
And I'm Adam. Today we're talking about mirror life, a possible new form of life built from mirror versions of the molecules found in all living things. It doesn't exist yet, but many of the mirror molecules we need to create life are already in labs today, and scientists believe it may soon be possible to create a fully functioning mirror organism.
The idea sounds scary. Yes, mirror biology can open the door to medical advances, but the creation of self-replicating mirror life goes way beyond that and poses risks unlike anything we've encountered before. To help us understand what's at stake, we're joined by Yensi Flores Bueso and Thomas Südhof.
Yensi is a synthetic biologist and co-chair of the Global Young Academy, a network institution of the Secretary-General's Scientific Advisory Board. She's wanted to be a biologist her whole life.
I was born a biologist. My mom used to make fun of me growing up because I would grab like the big ants and touch them. And she will ask, "What are you doing with that?" And I was like, "I'm tickling it." And I am really lucky to be from Honduras, which is such a biodiverse country, you know.
I have to admit, Yensi's ant story made me smile. We also spoke with Thomas Südhof.
My name is Thomas Christian Südhof. I am serving on the Secretary-General's Scientific Advisory Board.
In 2013, Tom shared the Nobel Prize in Physiology or Medicine with Randy Schekman and James Rothman for discovering how cells move and deliver essential materials. He also shared the story of how he received the news with us.
I got a telephone call and he basically told me that I had been awarded a part of the prize in 2013. I expressed, of course, my delight. And then I went on to the meeting.
And then you had a party when you came back?
We had lots of parties. Yes, it was a very, in many ways, wonderful time.
Our guests have spent years studying the molecules and processes that make life possible. Today, they'll help us understand what mirror life could mean and why it has so many scientists really worried.
But first, let's start with one of the fundamental features of biology that is key to the concept of mirror life. It is the very weird word chirality.
In biology, there is a principle that is called chirality. And that means that all the molecules in life have a handness to them. Like if you put your hands facing out and then you see your right hand and your left hand, they look identical. But if you try to put them one on top of the other, they won't match. So that's the principle of chirality. And very early on, life, when it started, decided to have left amino acids, which is the base of proteins, the unit of proteins, and had right-handed sugars, which is the base for all DNA. And that's how biology decided very early, and all life on Earth has this chirality.
Chirality matters because in all living things, molecules need to recognize and interact with other cells. For example, immune system molecules can only detect and attack dangerous viruses if their shapes fit together. In the same way, the molecules that help us digest food need to fit onto what we eat, like puzzle pieces. If the shapes don't match, they can't do their job. This is one of the basic rules of biology and has been how life on Earth has worked for billions of years.
Biologists like Yensi can now make synthetic versions of the key molecules, things like proteins, that are needed to eventually make life.
I will design this new protein and then we'll tell the computer to give me the DNA sequence that will make this protein, and then I will order it. The DNA will arrive by mail at my lab, and then I will use this DNA to transform bacteria. That's what it's called. So transformation is putting the DNA inside of the bacteria. And then the bacteria will make this new protein for me. And I will extract the protein from the bacteria and test it in the lab with different instruments to see if it does the work and it behaves as I want it to behave.
The ability to make synthetic molecules, like Yensi's doing in her lab, opens the door to making mirror versions of them also.
Now we need to understand the difference between creating mirror molecules that sit in test tubes or go into medicines and creating mirror life. which is very different.
Mirror life is the idea that you could create life where everything is exactly the opposite in terms of its handedness. So instead of having, as so to speak, only left-handed DNA, you only have right-handed DNA. Well, actually, there's no reason why that shouldn't work, as long as everything is of the opposite chirality.
Today, labs around the world can already create mirror cells. For example, they can create a mirror protein. These are exactly the same as naturally occurring molecules, but they just have the opposite chirality or handedness.
But what makes mirror molecules so interesting? What could they do that naturally occurring ones can't?
Think about a normal molecule in the natural world. It has millions of possible predators, other organisms that want to kill it, maybe even eat it. If the cell is in our body, for example, it might get killed off by our immune system. But a mirror molecule is essentially invisible to those predators. It's camouflaged because it has a shape that no one recognizes.
Because nature uses a different chirality, the enzymes or like the proteins in our bodies that do the work of coding proteins that are inside of us that are not ours are unable to detect them. So they're not caught. So they live there for a long time. And they're very stable in nature because nothing is able to digest it. Nothing is able to eat it or destroy it. So they last a long time. So this has very good advantages. Think of drugs, for example. A lot of the drugs that we take, we have to take a lot more because most of it won't ever make it to the target cell. But if we have this molecule, you can guarantee that we'll make it because it will last that long because our immune system won't get activated.
Mirror molecules might have some advantages for the development of medicines. They could stay longer in the body and therefore be more likely to reach the place where they are needed. Scientists think this could lead to longer-lasting treatments for diseases like diabetes, cancer, and chronic inflammation, meaning patients might need fewer doses and could benefit from more effective medicines.
So you could potentially have a drug that is in the body for days, weeks, months, not changed, not degraded, and against which there's no immune reaction. These advantages also, however, reveal the potential danger, because if you had a mirror organism, it is likely that it could invade any life form, plants, humans, animals, whatever, and they would not recognize it as foreign. They would not be able to digest it. They would not be able to do anything with it. They would be powerless.
This is where things could become dangerous, when we move from a mirror molecule that just sits there to a mirror organism that can reproduce and spread.
If you have a molecule in a test tube, that molecule won't be able to replicate itself. It won't grow on its own. But when we're talking about a mirror cell, then we're talking about an ecological entity, an organism that could potentially find food, and then it could reproduce and grow. These are cells that our body cannot recognize, so our immune system wouldn't be activated. We wouldn't be able to defend against them. We have nothing that can compete with it.
Yensi and Tom are talking about something that does not exist yet. A living organism made entirely from mirror molecules, known as mirror life.
If a mirror organism were ever created, it might not be recognized by our immune systems, antibiotics, or many of the natural defenses that protect us. In theory, it could spread through people and the environment in ways that would be very difficult, maybe even impossible to control.
I know of scientists that have told me, I cannot sleep at night. Think of this. So they are very worried. And that's why they came together and they are trying to raise awareness about the risks and trying to bring governance into this particular scientific endeavor.
One reason scientists are concerned is that there are no specific rules stopping the creation of mirror life. So far, the main reason it hasn't been created is because it's extremely difficult and expensive. But as technology improves, that could change.
Maybe we should discuss in the scientific community worldwide, including countries that are not necessarily always part of the conversation, about the need to have some degree of caution and the need to have some degree of regulation that will basically keep bad actors from going overboard and trying to do things that might cause incredible damage, not just to a few, but maybe to all.
And bad actors are not the only concern. Scientists are always trying to learn more and do things that have never been done before. As the science improves, someone may eventually create mirror life in a lab simply because it becomes technically possible.
And this is where the big risks could break out of the lab. A mirror organism that spreads across humans or our planet could pose an existential risk to us all. Because of this, a growing number of scientists are even talking about finding ways to pause or regulate research into mirror life.
It will take, they said, approximately 10 to 30 years, but with AI, that timeline might shorten significantly. This is moving forward very fast, and I think that timeline to be able to assemble something as complex as a ribosome might be much shorter than we expect.
Just a few years ago, an AI system solved a problem that scientists had struggled with for years, predicting the shapes of proteins, the tiny molecules that help make life work. Could something similar happen with mirror life? Could a new discovery suddenly make it much easier to create the key parts of a mirror organism? We don't know, but breakthroughs can happen faster than expected.
I'm optimistic that we will think about the consequences more, but I think in 5 years we have this window of doing this governance, anticipatory governance, where the risk is still not there. But it's clear that it can happen.
Yensi's idea of anticipatory governance is about putting in place guardrails before something dangerous happens, and it's harder than it sounds. We're much better at responding to risks after they've fully landed. There are all kinds of examples, from coal mining to smoking to fossil fuels. The rules we create to manage the risks usually come a long time after a lot of damage has been done.
But some risks are so significant that scientists believe we cannot afford to wait until they fully emerge. Mirror life may not exist today, but it raises important questions that we may need to address before it becomes reality. I asked Tom what advice he would give to the next generation of scientists.
I have several pieces of advice.
Oh, there you go.
So first of all, in response to a question like that, the usual answer is follow your vision, follow your dreams. It is a good answer, but I think it's also a bit disingenuous because in the real world, none of us can follow our dreams. We are bound by realities. So my advice is always to dream and dream broadly, but also think in your dreams also about what can be done potentially.
Young scientists will play a central role in that journey, and the decisions we make today could shape the future of this field for decades to come.
You've been listening to Science Forward.
Curious to learn more? Check out the show notes for links to the work of the UN Secretary-General's Scientific Advisory Board, and to stay connected with us on LinkedIn, X, and YouTube. Thanks for listening.